Hello, good morning, good afternoon, good evening to everyone across the world. Welcome to this week's presentation on global immunotalks, and I'm truly delighted to welcome this week's speaker, who is Professor Bana Jabri. Uh, Let me give you a brief introduction to Professor Jabri. She is the director of the Imagine Institute of Genetic Diseases, and she is also an endowed visiting professor at the University of Chicago. Before she took up this position at the Imagine Institute in Paris, which was last year, she served as the vice chair in the Department of Medicine and the director of the Digestive Disease Center and chair of immunology at the University of Chicago. Bana is trained as a physician scientist. She's an immunologist and an immunologist in Paris at the Necker Enfant Maladies and the Pasteur Institute, where she specialized in pediatric gastroenterology and human and mucosal immunology. Uh, Bana's work focuses on host microbial interactions, tissue immunity, and the mechanisms by which microbial signals contribute to the development of complex immune disorders and cancer. Her work has uncovered many key pathways driving tissue damage and the loss of immune tolerance in autoimmunity and inflammatory diseases, including celiac disease. Her work is truly elegant because she really combines deep insights in human immunology, being a physician scientist, but also with very beautiful, elegant mechanistic studies using sophisticated mouse models of disease. For this work, she has received several prestigious awards, including the International W.M.K. Warren Jr. Prize in Celiac Disease, the Quantrill Award for Excellence in Teaching, and the Lloyd Mayer Award in Mucosal Immunology. She is also an elected member of the Association of American Physicians. So it gives me great pleasure to welcome Bana to this week's Global Immunotalks. So Bana, as is customary, I'd like to ask you a very special question. And this week's question is, let me see if I can find this week's question. Let me see, where is it? I think it's in the chat box here. Here it is. What advice would you give about setting long-term goals whilst managing the pressures of short-term milestones?
Important question for all of us. So I would first say it's important to address important questions. I think one shouldn't shy away and one shouldn't be afraid of really addressing questions when things are impactful and will make a real difference. I think we are here as scientists and physicians to make a difference and not just to publish papers. And so I would say the key is first to think what are fundamental questions, what are things that will change our way of thinking in the future. And then one has to be also, of course, strategic, because if you ask an interesting question, but you don't have the means to answer it, it's not worth asking the question. So one has to develop also the tools and make sure that one can have either available tools or develop the necessary tools to address the questions one wants to address. And then if one asks a question, I think one can also have an idea that when one addresses this question, one can go, maybe one part of the path halfway and then full way. And actually, as we do science, we always realize we never go the whole path because each time we answer a question, there's a new question that arises. So what I would encourage young people to do is not to be afraid. And to say that if they work hard, they address key questions, and then they have the right tools, they will do well. And so that would be my main advice.
Thank you so much, Bana. It's beautiful advice to everyone, not just young people, even not so young people as well. Thank you so much. So before you begin, I should also mention a couple of things. Firstly, we're very proud to say that today is the inaugural session where Global Immunotalks is being relayed directly on the YouTube channel. Those of you who've joined Global Immunotalks previously will remember that it was through a Zoom account and then subsequently it was posted on YouTube. Here, Bana's talk is being relayed directly live on the YouTube channel, so that's a pretty exciting innovation. And secondly, I'd like to remind you that there will be no Q&A, live Q&A session, but if you do have questions, please feel free to ask this on the YouTube channel. And I believe there's a QR code that's going to be on the screen shortly where you could access this. And finally, let me also say next week's speaker will be Professor Mark Jenkins from the University of Minnesota. So, Bana, without further ado, I welcome you to the podium to give us your presentation. Look forward to it.
Thank you so much, Bali, and thank you for all those involved in the organization of the IMMUNOTALKs. So I'll share my screen, and I will go in for full screen. Can you see my screen?
Yes, we can.
Okay. So, oh, sorry, I lost it. Yeah. First, I would like to say that, the picture on the left describes me jumping from the United States, the University of Chicago, to Paris, but I still have a position at the University of Chicago, which I'm very grateful for. But I just wanted to tell everybody you have my email address, both the French and the American email address on the screen. And I wanted to say that the Imagine Institute and France are hiring faculty. So if you are interested, please don't hesitate to email me. It's even beyond immunology. And also I'm just mounting my lab at the Imagine Institute. I just started this January. So if there are postdoctoral fellows interested, so please don't hesitate. So I'm going to talk about decoding dysregulated immune responses to dietary antigen, which, as we know, cannot have only an impact on diseases such as food allergy or celiac disease, but potentially Crohn's disease and other disorders. I'm going to have the string, and for some reason I cannot forward my. Oh.
Do you want to maybe unshare your screen and then reshare it?
Yeah, I will do that. I will reshare. And yeah, wait a second. Right. I'm sorry about that, because we had checked
That's
and
okay.
it was working. And
Yeah.
can you see it?
Yeah, I can see it.
Yeah. Okay,
Yeah.
now it's working for whatever reason.
Great.
Okay, so the question I would like to discuss with you is a little bit how can we come to dysregulated immune responses to dietary antigens? But another question that I find extremely intriguing is that we have a rise in all types of complex immune disorders, such as diabetes, psoriasis, celiac disease, food allergy, and yet we know that these different diseases are mediated by very different types of immune type of T helper T cells. You have for psoriasis TH17, for celiac disease TH1, for food allergy TH2, and we still poorly understand why a given individual will not only develop a complex disorder, but why a given individual will develop a disorder that will be more TH1 or TH2 mediated. And this is in particular interesting in the context of, for example, dysregulated immune responses to wheat, because you have as well wheat allergy as celiac disease, and both of them are mediated by very different diseases. Celiac disease being much more comparable to organ-specific immune disorder. And in that sense, what is interesting in the context of celiac disease is that there is a very strong MHC class 2 association, like for organ-specific immune disorders. You have development of autoantibodies against tissue transglutaminase, and in particular, what distinguishes an autoimmune disorder from an inflammatory disorder, you have the specific destruction of one type of cell, which is the surface epithelial cell, that leads to villus atrophy. And the cells that mediate this destruction are intraepithelial lymphocytes, but they require the activation of gluten-specific CD4 T cells. So in this first part of the talk, I'm going to discuss, in the context of celiac disease, how can we think about the induction of T helper 1 immune-mediated disorders. So what's interesting about gluten is that it is very poorly digested because it has a rich content in proline. And the other interesting factor about gluten is that it is an excellent substrate for an enzyme that is ubiquitously expressed that is called tissue transglutaminase type 2. And this enzyme will promote deamidation, introducing negative charges into gluten, which has barely any negative charges. It turns out that the two predisposing MHC molecules, DQ2 and DQ8, have positively charged pockets. And so this introduction of negative charges will render gluten peptides highly affine. And the presence of proline that rendered the gluten poorly digested will generate peptides of sufficient length so that they can bind to this MHC class 2 molecules. So the work of Chetan Khoshla, Ludwig Solid, and Fritz Koenig in that field explain very well how you can actually mount an immune response against gluten, but they don't. But it doesn't explain why you would mount a TH1 mediated disorder and not, for instance, regulatory T cells, as we know that in the gut, the presence of TGF-beta and retinoic acid really favor the differentiation of regulatory T cells. So one thing that is interesting when you study organ-specific autoimmune disorders is the cytokine interleukin-15, which is actually overexpressed in a number of autoimmune disorders such as psoriatic arthritis, alopecia, and rheumatoid arthritis, as well as type 1 diabetes, and in some cases, inflammatory bowel disease. And so we became interested, Valerie and William DiPaolo became interested in asking the question, could sterile inflammation, as seen in the context of upregulation of IL-15, be actually responsible for the induction of a TH1 response to dietary antigen? So as I already said, we know through the work of many people that include Musida, Sherwood, but Fiona Powie, Yasmin, and many others, that retinoic acid and TGF-beta will promote the differentiation of regulatory T cells. So what Valerie and William found is that in the context of IL-15 overexpression, now what you can see is that you will have the induction of an inflammatory phenotype in dendritic cells that promotes a TH1 response. And the reason this is seen, again, I have a problem. I think it's linked to this. I'm really sorry because... and I cannot... So in this context, what they were able to show is that normally if you have retinoic acid and you increase the dose of retinoic acid, you will increase the number of regulatory T cells. But if you have IL-15 present, the more retinoic acid you have, actually the less regulatory T cells you have, and the more TH1 cells you induce. And one pathway that's responsible for that is the fact that in presence of retinoic acid, you actually activate the MAP kinase pathway, such as JNK2, and the activation of the MAP kinase that is only induced when both retinoic acid and IL-15 signal into dendritic cell will render those dendritic cells pro-inflammatory, promote the induction of IL-12 that then induce TH1 cells. So you can see that even retinoic acid that is seen as a tolerogenic factor will become pro-inflammatory in a context where IL-15 is overexpressed. And by the way, retinoic acid is also used in the context of fight against cancer. But in addition to IL-15, there's also the concept that viral infection could actually promote autoimmune disorders, and several epidemiological studies have linked the development of celiac disease with viral infection. And this is actually also the role of environmental factors is also suggested when you look at the prevalence of the disease, function of wheat consumption, and the expression of the predisposing HLA molecules. You could say that there is overall a correlation, but there are striking outliers, such the region of Karelia, where in Finland you have less than 0.3% of individuals who develop the disease, and in Russia, where more than 3% of individuals develop celiac disease. And so this really points to the fact that there are other factors than gluten that play a role in the development of celiac disease, and that, for instance, microbes could play a role. And in that regard, it is also interesting to note that the predisposing genes that have been identified through GWAS studies are actually also genes that are involved in the protection against infection. And so the concept that infection could be part of a reason for developing intolerance to gluten is certainly a possibility. So when we think about the role of microbes in autoimmunity and in dysregulated immune responses to dietary antigen, one usual way of thinking about it is to put forward molecular mimicry, meaning that somehow there is a cross-reactive T-cell response to the microbe and to either the self-antigen or the dietary antigen. Such possibilities have been evoked and also shown by Fritz Koenig and Jamie Rostron, but one would have to invoke each time that a given virus or a given microbe needs to promote also a cross-reactive response. The other model that is often proposed is bystander activation. But here the problem is, if you really had bystander activation, you would activate a number of times dysregulated immune responses, because each time you would have an infection or any assault that would activate one cell, it would activate other cells. So the lack of specificity here and of checkpoints poses a real problem. So to address this question, we felt that what was important was to develop a model in which we would have viruses from the same family, and one that would promote immunopathology and one that would not. And so we really thought about different types of models, and we wanted also a model that would be relevant to human disease, because the virus would also infect humans, and also not to have to use neonates like for rotavirus, but also to be able to do it at different age stages. And so in collaboration with Terry Dermody, who is now at Pittsburgh University, we went and screened different reoviruses that are double-stranded RNA viruses that can create easily reassortants. So this allows to actually look at the individual role of different genes and ask the question of could we identify a virus that would induce a loss of all tolerance, and in particular TH1 responses, and one that would not, and have both viruses actually infect the gut. And so by screening different viruses, we identified a T1L that was known to infect the gut, but the reassortant T3DRV, in which the S1 and L2 genes from T1L were introduced, and that allowed T3D to infect the gut similarly to T1L. And then the mice were infected, and we would look in particular at the response to dietary antigen. And this work was led by Romain Bouzia, who was a staff scientist at Genentech, and Reinhard Hinterlein, who is an assistant professor on the tenure track at the University of Pittsburgh. And so, as many of you know, Pierce patches are viewed as the inductive side of protective immunity, and this has certainly been shown for T1L, and T3DRV, I'm going to show you, also infects that, whereas mesenteric lymph nodes are thought to be the inductive site for all tolerance, because if you remove mesenteric lymph node, you have a lack of induction, for instance, of regulatory T cells to dietary antigen. As you can see here, Both viruses induce antibodies, high levels of antibodies. What's interesting is that T1L induces somehow higher levels than T3DRV, but both clear the virus. I'm not showing it here at the same time, around 6 days post-infection. This was interesting. I'm not going to show you the data. We used the fact that T1L induces more antibody titers to then go into celiac disease and show that what I'm going to illustrate here in the mouse was actually also relevant in the context of celiac disease. And so both viruses, T1L and T3DRV, infect the Peyer's patches, and both will induce similar levels of TH1 cells in the Peyer's patches. Here we are looking at the host response, and in the context of the Peyer's patches will not, in fact, impact the host regulatory T cell response. We did a RNA-seq analysis of the epithelium, the lamina propria, Peyer's patches, and mesenteric lymph node. And in an unbiased manner, you can see that analysis of the RNA-seq allowed, using a spanning tree, to really actually differentiate the responses in the epithelium, lamina propria, Peyer's patches, and mesenteric lymph node. In white, you have sham-infected cells. You see that the red and the blue distinguishes themselves from the white, the sham-infected cells, but you can see that the red and the blue are intertwined, meaning that both T1L and T3DRV induce transcriptional responses in Peyer's patches, but that cannot be distinguished from each other, really indicating that both of those viruses induce similar responses, protective responses, and transcriptional responses in Peyer's patches. What is interesting is now when you look at 48 hours, at 6 hours, you cannot see any difference in the lamina propria and in the mesenteric lymph node between sham and infected mice. But at 48 hours, you can see a clear distinction between the sham-infected mice and the virally infected mice. And what you can also see is that the blue is closer to the white, meaning that T3DRV induces less differentially expressed genes than T1L, both in the lamina propria and in the mesenteric lymph nodes, which we know are sites where the response to dietary antigens can be altered. And I'm summarizing this data because they have been published. So both viruses will induce similar immune responses in Peyer's patches, but only T1L will alter dendritic cells and promote TH1 responses to dietary antigen, whereas T3DRV is unable to do so. And we were able to show similar results using norovirus, CR6, and CW3, where only the acute virus was able to induce a TH1 response to dietary antigen, but not the chronic virus, norovirus. And importantly, I stress that if you infect the mice with salmonella or leishmania, you will not induce a TH1 response to dietary antigen, meaning that it's only very particular microbes that can induce this response. And even within the same family, certain viruses will be able to do it, but not others, which really poses the question of what the underlying mechanisms are that endow a given microbe to induce this dysregulated immune response. So Elaine Kwame was a PhD student in the lab, and she really tried to tackle that question. So again, just because of time, I'm going to summarize data, but I think an important concept that I would really like to stress is that in the context of dendritic cells, what you can see is that even if a given dendritic cell, like I'm looking here at CDC1s, expresses retinoic acid, which normally is associated more with tolerance. These dendritic cells also have high levels of interleukin-15, and these dendritic cells also express TLR9. So if you were to put CPG, this dendritic cell would actually induce IL-12, would actually produce IL-12, and could promote inflammatory responses and not just regulatory responses. If you look now at the CDC2s, they have TLR4 and TLR6. So if you put LPS on this dendritic cell, it will activate NF-kappa B. If you put LPS on CDC1, they will be unable to activate NF-kappa B because they don't have the receptor for LPS. So there is really strikingly I would say, division of labor or a division of sensing between the different dendritic cell subset. And I think this is very important to keep in mind. What is very particular about the CDC1s that can present dietary antigen, and in collaboration with Daniel Musida and Gabriel Victor using lipstick in the context of the viral infection, we could really show that both CDC1s and CDC2s can present dietary antigen to T cells. But what is very particular to CDC1s is that they are able to uptake epithelial cells. That's work of also Julie Blunder, and we were able to confirm that. And also the CDC1s are the only cells that express CLEC9A that is able to recognize necrotic cells. So you see that there are two types of dendritic cells, both able to present dietary antigen, but with very different capacities and different innate sensing receptors. We are all aware that the type of cell death is very important. Apoptosis is often associated with no inflammation or even anti-inflammatory pathway, whereas necroptosis or different types of non-apoptotic cell death are associated with inflammation. And so what Elaine did is she used MLKL knockout mice to address the question of the role of necroptosis. And as you can see, in absence of MLKL, there is a significant decrease in the capacity of the mice to develop a TH1 response to dietary antigen. She then did a bone marrow chimera to ask the question whether necroptosis was important in the hematopoietic compartment or the non-hematopoietic compartment. And as you can see here, the clear response is it's in the non-hematopoietic important that necroptosis is important. And here, knowing that reovirus can infect epithelial cells, likely it is necroptosis in epithelial cells that plays an important role. Now, the ability of CDC1s to express CLEC9A, knowing that CLEC9A is actually a receptor that can recognize necrotic cells because it recognizes F-actin, and again, its expression is restricted to CDC1s, posed the question, knowing that also necroptosis was important for the induction of TH1 response to T1L, whether CLEC9A was important. So Elaine used both genetically modified mice, but also I'm not showing it here, an antibody that blocks CLEC9A. And in both cases, she could show that there was a significant decrease in TH1 responses to dietary antigen. And that could also be confirmed not just to OVA, but also to gluten-specific T cell responses. I want to stress, I'm not showing the data here, that in both cases, CLEC9A and necroptosis in the MLKL knockout and CLEC9A, there was no impact on the blockade of regulatory T cells. So here I'm just going to summarize the data. What Elaine was able to show is that when you have T1L infection, you have a blockade of regulatory T cell. This can be associated to the M1 gene that is present in T1L. And this blockade of regulatory T cells was dependent on type 1 interferon, but it could be either signaling into innate cells or into T cells, because only blocking of type 1 interferon that blocks both pathways was able to restore regulatory T cells, whereas when we were using OT2 T cells that lack the interferon receptor, you would still see Treg blockade. In the context of TH1 responses, here there was not only an involvement of the M1 gene, but also of the M2 gene in T1L. And importantly, what was required in addition to type 1 interferon, where type 1 interferon were only necessary for Treg blockade, you also needed to recognize necroptotic T cells and have necroptosis in order to induce this Th1 response, really showing that the induction of actually the pathogenic response required many more dysregulation and impact on different checkpoints, whereas the blockade of regulatory T cells, it was sufficient to induce type 1 interferons. So as you can see here, no molecular mimicry was required. And also one cannot talk just about bystander activation because it required very specific receptors expressed on the cells that were able to present the dietary antigen. So this, I think, in some ways really shows that you can be specific and that you don't need to invoke a molecular mimicry in order to mount dysregulated immune responses. The thing I would really like to go back to and insist on is, again, that blocking regulatory T cells is not enough to induce pathogenic T cell responses. And this is very clearly shown here. It was published in the initial paper by Bouziat et al., but we confirmed it again. If you give poly-IC to mice, you will block the induction of regulatory T cells, and this is dependent on interferon receptor signaling, and you can do that in vitro as well. But poly-IC by itself will never be able to induce the Th1, and I shouldn't say never, it will not induce a Th1 response, and whereas T1L can, really indicating that type 1 interferon can block Tregs, and blocking Tregs is not enough to induce a Th1 response, and again, that T1L activates these other pathways that include necroptosis and recognition of necroptosis. I think this concept that one can dissociate blockade of Tregs from induction of pathogenic T cell responses was also nicely shown by the paper that was recently published from Daniel Musida's group by Maria Canesso, and where they could show that upon helminth infection, the dysregulation in the ratio of the different dendritic cell subset would lead to the blockade of Treg, but was not enough to induce Th2 responses. And in the context of celiac disease, for the moment, nobody was able to show that there were regulatory T cells against gluten. And again, this does not mean that you will have a pathogenic response. And I'm not showing you the data, but in our different genetic models, we could dissociate blockade of Treg and induction of pathogenic Th1 responses. So this again poses then the question of how can you induce Th2 immune responses and what are the requirements for that? And this is work by a very talented MSTP student, and again in collaboration with Reinhard Hinterleitner, who is, as I said, the assistant professor at the University of Pittsburgh. So TET2 is a methylcytosine deoxygenase type 2 that is involved in active demethylation. The concept of active demethylation was actually first introduced by Ron Schwartz when he was looking at IL-2 regulation. And he proposed that active demethylation could exist, which was highly controversial at the time. But with the identification of the TET enzyme, people were able to show that you can have actually active demethylation. And this TET enzymes, as well as DNMT3, which are methylation enzymes, are associated with the development of leukemia, and in particular with a CHIP, which is clonal hematopoiesis of undetermined origin. So Maëlys Meisel was also a postdoc in the lab, and Maëlys and Reinhard combined their forces to address the question of how a pre-leukemic myeloproliferation could be induced in the context of TET2 deficiency. And they were able to show that IL-6 and the presence of bacteria such as lactobacillus that translocated in the context of TET2 deficiency were actually required and sufficient to promote pre-leukemic myeloproliferation in the context of TET2 deficiency. And that's because GMP, in the absence of TET2, overexpress IL-6, and this renders those cells hyper sensitive to IL-6. They then proliferate, which provides them, by the way, also a selective advantage, and then sets the stage for pre-leukemic myeloproliferation. They were also able to show that giving TLR2 ligands was actually sufficient to promote this pre-leukemic myeloproliferation in TET2 deficient mice. But what was very interesting is, as I said, bacterial translocation was essential, because if you gave antibiotics, there was no pre-leukemic myeloproliferation. But if you gave TLR2 ligands or lactobacillus, you could induce pre-leukemic myeloproliferation, but you were not able to induce barrier dysfunction. So this really posed the question of what was the microbial signal that was responsible for barrier dysfunction in the context of TET2 deficiency. So when they looked at the impact of TET2 deficiency in the gut, what was very interesting is that the site where you had the most differentially expressed genes was in the jejunum, neither in the ileum, nor the colon, nor the duodenum. And when you look at the sets of genes, I'm sure you can identify IL-25 and the receptor for IL-25. And when Kishan analyzed the RNA-seq data, really the pathway that came up was the pathway that was associated with differential expression of Tuft cells. And as you can see, TET2 deficient mice have a very significant increase in Tuft cells. So we know through the work of Loxley group, Van Moltke, and also Wendy Garrett and others, that Tritrichomonas is a protozoa, a commensal protozoa, that can induce a very interesting circuit whereby this protozoa, through the production of succinate, can activate Tuft cells that produce IL-25, that then activate ILC2s that produce IL-13, that then in turn further act on Tuft cells and expand them. But what is very important to remember is that this circuit does not involve T cells. This circuit involves ILC2s. And this is actually really interesting, because this means that this protozoa in this context, in the wild-type context, is unable to promote T cell responses. The other very important concept that was shown, and you can find it in papers from Wendy Guides actually in supplemental data, is that even though the protozoa is never cleared by this circuit, what you observe after a few weeks is that the IL-25 response is reduced and disappears. So it's a transient response. So this protozoa increases a transient IL-25 response that is associated with Tuft cell expansion. So when Kishan looked at the TET2 deficient mice, what was striking is, like wild-type mice, Trichomonas was never cleared. But the big difference was here. When you look at IL-25, wild-type mice, as was previously shown, IL-25 is increased in wild-type mice, but then it goes back to normal levels. In contrast, in the TET2 deficient mice, even 3 months, and Kishan looked at 6 months, IL-25 is still maintained at high levels. And this is associated with alteration in barrier function, as shown in this barrier function, in the decrease of this barrier function gene. So the question then becomes, in the context of this barrier dysfunction and this chronic IL-25 activation in absence of Trichomonas clearance, can one invoke a T cell response? Because the microbe is still present, meaning it could still engage T cells chronically. And so Kishan looked at T cell responses in the context of Trichomonas infection in TET2 deficient mice. And as you can see, indeed, he could see in the lamina propria an expansion of GATA3 positive, FOXP3 negative cells. And interestingly, these T cells were activated, and this activation was dependent on the presence of Trichomonas, because when you give metronidazole, which, by the way, clears Trichomonas, you decrease CD69 expression. There was also a correlation between IL-25 expression and the number of TH2 cells. And most importantly, when Kishan gave anti-CD4 depleting antibodies, IL-25 disappeared and came back to the levels of wild-type mice. And this was then associated with a normalization of barrier function. So what I showed you here is that in the context of TET2 deficiency, Tritrichomonas is able now to induce a TH2 response. So let me take a step back and let's rethink about how TH2 differentiation occurs. And there are several features that are very unique to the TH2 pathway. One feature is that the cytokine that is also an effector cytokine, because one of the prototypic TH2 cells are IL-4 producing cell, the same cytokine is a cytokine that can promote TH2 differentiation, which is not at all the case for TH1 cells where IL-12 can play a role, or for example, TH17 cells where IL-6 and TGF-beta play a role. So the effector cytokine can also be the differentiating cytokine. The other important difference is that for TH1 differentiation or TH2 or TH17 differentiation, the production of cytokines by the dendritic cell producing the dietary antigen is important. But in the context of TH2 cells, the recruitment of innate cells such as NKT2, basophils, and other innate cells is key. IC2s is key actually in promoting the TH2 differentiation, meaning that the checkpoint that interferes in the context of TH2 cells involves this recruitment of innate type cells. So let's go back to TET2 deficiency. What you can see here is that if you do now an in vitro differentiation using anti-CD3 and anti-CD28 and IL-2, meaning under non-polarizing condition, what you can see now is that those naive T cells activated under non-polarizing conditions can produce IL-4 and can become TH2 cells, and that the differentiation into TH2 cells is dependent on IL-4. So this data suggested that in absence of TET2, now the naive T cells are able to produce IL-4 and promote the production of IL-4 by the naive T cells, can promote their differentiation into TH2 cells. If this is true, then one should be able to test that in vitro. So if you use wild-type cells and then knockout cells that produce TH2 cells, and again, in absence of IL-4, they will not become TH2 cells, which mimics what I showed you before. But then they designed with Morgan a assay where they took cells that were reporter for IL-4, so wild-type cells that were reporter for IL-4, and they incubated them, they co-incubated them either with wild-type cells or with knockout TET2 cells. If the TET2 knockout T cells produce IL-4, and this is sufficient to promote TH2 differentiation, even in wild-type cells, now we should see an increase in IL-4 GFP positive cells in the wild-type cells. And that's exactly what they saw, really further supporting the hypothesis that in the absence of TET2, IL-4 production by T cells, by naive T cells, under non-polarizing condition, can promote TH2 differentiation. So to take it a step further, this really suggested that it was TET2 deficiency in T cells themselves that may be key in inducing a dysregulation of IL-4 production. So Kishan used TET2 deficient they used CD4 Cree mice, meaning that this time TET2 was selectively depleted in T cells. And as you can see, you will still see in mice the differentiation of TH2 cells in presence of Tritrichomonas. What was interesting is when now analyzing the naive T cells, there were a number of differentially expressed genes, and many of those genes are actually associated with the regulation of T cell activation and with genes that are implicated in the regulation of TH1 versus TH2 differentiation. And the pathways that were enriched had to do with IL-5 and STAT5, interferon gamma, and type 1 interferon. So looking now more carefully at the epigenetic state, what Kishan could see is that there were a number of differentially methylated loci in the naive CD4 T cells lacking TET2. And again, the transcription factors you can see here are involved in the decision of TH2 versus TH1 differentiation. Using ATAC-seq, they could see that these hypermethylated sites were also associated with differences in accessibility. And again, here looking at ATAC-seq and methylation sites, what I really want to point out is the unstimulated state, where you can see that all these changes are already present under unstimulated conditions, meaning in the unstimulated state, suggesting that the cells are really poised to respond differentially upon TCR activation and then really induce these TH2 sites. And when you look at the transcription factor binding sites that were altered, again, RANKS1, ETS1 are genes that are associated with differential differentiation between TH1 and TH2 cells. So does this TET2 deficiency in lymphocytes have implication for immunopathology? What Kishan could see is that indeed those mice developed a much more food allergy using low doses of cholera toxin. He was even able to see in one mice development of food allergy even in absence of cholera toxin initially, but then he didn't want to keep on doing many mice, so he used low doses of cholera toxin. And this induction of food allergy was dependent on IL-25 because giving anti-IL-25 prevented induction of food allergy. And again, conversely, giving recombinant IL-25 was enough to induce barrier dysfunction. And so here, what we believe is happening is that in an IL-25 dependent manner, the response to Tritrichomonas and this TH2 response will be inducing barrier dysfunction associated with increased food allergy. So I want to go back to this notion that all the difference we were able to see were under non-polarizing condition. And I think this is extremely important because I think also in the context of the work from Anjana Rao's lab, they used polarizing conditions when they looked at TET2 deficient T cells. So the difference we see is when we use a non-polarizing condition. When we now use polarizing conditions, you see that there's no difference in the ability to differentiate into TH2 cells between wild type and TET2 deficient mice. And importantly, if you now use a helminth that can induce IL-4 responses for innate cells, again, you see no differences between wild type and TET2 deficient mice, meaning that the only time you are able to see this response is in the context of a commensal protozoa that likely is unable by itself to induce a TH2 response. So this would suggest that if now we give IL-4 to wild type mice, in the context of Tritrichomonas infection, we should be able to induce a TH2 response. So as you can see, if you just give recombinant IL-4, this is not sufficient in wild-type mice to induce a TH2 response. If you only give Tritrichomonas, that's not enough, as previously shown. But now, if you give Tritrichomonas and recombinant IL-4, now you can see the differentiation of TH2 cells that is accompanied by an upregulation of IL-25, really showing that in the wild-type context, this protozoa cannot induce IL-4, therefore rendering the requirement for T cells to produce their own IL-4 in order to differentiate in TH2 cells. Now, the relevance to human disease, if you now take this data and the pathways that were enriched in the TET2 deficient mice in the naive T cells, and then compare that to the pathways that were identified also in naive T cells of allergic patients, activated for 72 hours under non-polarizing condition, we had a very significant overlap between the pathways identified in the TET2 deficient T cells of TET2 deficient mice and the pathways identified in allergic patients. It's interesting to note that in tall patients, they develop allergic responses to medication with time. And when one analyzes the patients that develop these allergic responses, the key gene that is differentially expressed is RUNX1, which is the top transcription factors identified to be differentially expressed in TET2 deficient T cells. So what I have shown you here is that in the context of helminth infection, where you can activate innate cells to produce IL-4, you will induce a TH2 response in wild-type mice or in TET2 deficient mice with no differentiation. However, if you're now in the context of a commensal protozoa that is unable to activate this TH2 response, this IL-4 production and the recruitment of innate cells, this is now based on ILC2s, and there are key checkpoints such as A20, as shown by Loxley, that somehow regulate that pathway and make them transient. And so you will not have chronic TH. You will not have the ability, A, to induce a TH2 response, and B, you don't have the ability to induce a chronic circuit. Now, in the absence of TET2, where you have IL-4 production, and now you can induce a T cell response. And because Tritrichomonas is neither cleared by ILC2 nor by TH2 cell, in presence of this chronic presence of the protozoa, you induce a chronic TH2 response that is associated with Bayer dysfunction and allergy. This is the last slide. I just want to make a point that I think is really intriguing. It turns out, as you know, that black 6 mice are more associated with TH1 responses and bulbous C more with TH2 responses. We analyzed, and others have done it also, naive T cells, and we could see the same phenotype then in TET2 deficient T cells, meaning that naive T cells from bulbous C can produce IL-4 under non-polarizing conditions. But what is very interesting is that bulbous C mice lack actually the receptor that can recognize succinate and can activate the circuit in presence of Tritrichomonas. So it's as if there could be an evolution in bulbous C mice that have IL-4 to lack actually the receptor to mount these chronic TH2 responses. And in some ways, this reminds me a little bit of what has been shown in a very different context by Lewis Lanier and Yokoyama in another context, where they, as you know, LY49H recognizes CMV, and they were able to see that it was a molecule induced by the virus that was recognized by the activating NK receptor that allowed to clear the virus. But Lanier then said that doesn't make any sense. There must be, the virus would not produce something that will allow its clearance. So he went and looked at other mouse strains, and then he found that actually the same ligand was recognized by inhibitory receptors in these other mouse strains. So I think this is again intriguing. It doesn't prove directly something, but it is intriguing to know that in the context where you have the capacity to mount a TH2 response against Tritrichomonas, the circuit cannot be induced. So again, just in summary, I think when we think about how do we induce these pathogenic responses, one has to take into account the host and the microbe. And not only the microbe, but as I have shown for the viruses, one has to go at the strain level in order to really understand the mechanism. And as you can see, there are many checkpoints that regulate the induction of pathogenic T cell responses. And I think if we want to understand the development of these diseases, it's not just about understanding how Tregs are blocked, but also about the pathways that promote these pathogenic T cell responses. So again, I think I have thanked really the people in the lab that have done that: Kishan, Morgan, Hope, Veronica, and Lee for the TET2, and then Elaine, Kwame, and Nick Asher, and others for the virus responses. And a big thank you to previous postdocs who are extremely successful assistant professors, hopefully soon tenured, Reinhard and Marlies Meisel, and of course, thank you to Terry Dermody for the collaboration on viruses, and NIH and Helmsley for funding. Thank you.
Thank you very much, Bana, for that very beautiful, elegant talk and work. So this concludes today's presentation. As I mentioned, there will not be a live Q&A, but if you do have questions, feel free to post this on the YouTube comment section, and Bana will get to answering your questions when she can. And just to remind you, next week's speaker will be Dr. Mark Jenkins from Minnesota. So this concludes this week's Global ImmunoTalks presentation. Thank you all for joining and have a nice day. Take care. Bye.